Today we're going to be talking about enzymes. So title in your notebook can be enzymes. All right, so what is an enzyme? An enzyme is an example of a protein molecule that speeds up chemical reactions. We know that protein molecules have C, H, O, and N, and they help build up the structure of things. Enzymes are one example of a protein. And so enzymes aren't necessarily needed for reactions to occur, but they help them occur a lot faster. In this picture below, we see that there's this pinkish red molecule. It's connected together. If we wanted to separate this pink molecule, we could, and it would take a little bit. But if we have this enzyme here, this blue piece, this enzyme can help break it apart a lot faster. So enzymes build things up or break them down. We've learned about some enzymes. Helicase unwinds DNA. Polymerase makes a new strand of DNA. We have some examples down here in pictures. In this example, we have two of these different colored circles, and they're going to be connected together with the help of this orange enzyme. So that would be building something. In this picture, we've got a dark blue molecule, and it's broken apart into two things using this enzyme. Now, these are cartoon pictures, right? We don't have colored circles in our body. These are made up of molecules, carbons and hydrogens and oxygens and nitrogens and whatever atoms they might be made up of. So enzymes often end in ase. We've learned about a few, right? Helicase and polymerase, those help with DNA. Lipase we'll learn about, digests fats, lipids. See that word lipid in there. Amylase is in your mouth, secreted by your salivary glands, and helps break down carbohydrates. So if we take a look at this graph, my question up here says, what happens to an enzyme if the temperature gets too low? So enzyme activity and then temperature. When my temperature is really low, my enzyme activity is also really low. When my temperature is really high, my enzyme activity is also really high, or really low, sorry. So it works best at this middle temperature. Now for each enzyme, this temperature that it works best at is going to be different. But too low of a temperature or too high of a temperature is going to cause the enzyme to not work as well. Think about if you wanted to, run a marathon or do 100 push-ups. If they put you in a room with zero degree temperature, it's probably going to take you a little bit longer. Likewise, if they put you in a room, this is in Celsius, but let's say 110 degrees, it's probably not the optimal temperature for you to run a marathon or do 100 push-ups. So the middle temperature works the best. So enzymes require a specific temperature to work, and for each enzyme that might be different. One enzyme might work best at 70 degrees, the other might work best at 30 degrees. Now, pH is a measure of how acidic or basic an environment is. So if we look, here are two enzymes. Pepsin, we learned about. Pepsin's in our stomach, and it helps digest proteins. So at what pH does pepsin work the best? Well, notice the activity is highest around 3. 0 to 7 is acidic. So 3 is pretty acidic here. So enzymes require a specific pH, an acidic or basic environment, to work the best. Again, they'll work at other pH levels, but there is an optimal or best pH level that they work at. So if we take a look, the pH of the stomach here, where pepsin works, is about 2. Notice, Pepsin works pretty well at a pH of 2. So you'll often find that the environments where these enzymes are found have the right conditions for them to work well. Trypsin, on the other hand, works well at a pH of 8, more basic. So what we're going to write down is that stomach acid provides an acidic environment for enzymes to work properly. So enzymes are in a lot of places in our body. They're in our mouth, they're in our stomach, they are in our intestines, and each of those environments has a different pH and a different temperature. And there are enzymes in those different environments that are suitable for those specific temperatures and pH levels. So in the case of pepsin that digests proteins, the stomach acid provides the perfect acidic environment for that pepsin to work properly. So if we take a look, trypsin is found in the small intestine. And in the small intestine, the pH is around 7 or 8. And so you'll notice that, hey, trypsin works the best around a pH of 8, which makes sense because it's found in our small intestine. So bile is released by the liver, and the bile actually helps make the correct pH for enzymes in the small intestine. So the big story here is that different parts of our body have different temperatures and different pH levels. And enzymes there work well because of those pH levels and those temperatures. So bile helps make the environment suitable for trypsin. Stomach acid makes it more acidic so that pepsin can work really well. So examples of enzymes. We have amylase. Amylase digests carbohydrates. So starch is broken down using amylase. This is secreted in your saliva. You can also find it in the duodenum or the small intestine as well, but the one we talked about is in your saliva. We have lipase, digests fats. You see the word lipid in there. Remember, lipids often look like an E. They're made up of fatty acids and glycerol. I know this says monoglyceride. Pretend it says glycerol. But lipase digests fats. Fats are digested in our duodenum, in that first part of the small intestine. Now, the difference between lipase and bile, remember, bile emulsifies fats. So bile breaks them down into little tiny droplets. Lipase breaks them into their fatty acids and glycerol. Okay, so you definitely need to know the difference between these two. So if you want, you can write down that bile emulsifies fats. Lipase breaks down fats. And remember, emulsify just means it takes this big glob and breaks it into smaller globules. We have pepsin. Pepsin digests proteins in the stomach. And then renin, which we're looking at in our lab, curdles milk. And this is found in the stomachs of newborn mammals. You don't secrete renin anymore or produce renin as an adult, but as a baby, you produced renin to curdle the milk that you drank from your mom or from formula. All right, last thing we're going to talk about here is how exactly do enzymes work? There's a couple different models. If you take a bio class later on, you'll learn more about these different models. The one we're going to talk about is the lock and key model. And this explains how enzymes work. They work kind of like a lock and a key. So we have some words we're going to talk about here, but let's just take a look at this picture. So we have a key and it fits into a lock. And if it's the correct fit, right? Something's going to happen. We can open that door. If it's the wrong key, it's not going to fit and it won't open that door, or no reaction will happen. Enzymes are similar. Let's take a look down here. This is like our key fitting into our lock. If it's the correct fit, then our reaction will happen and things will be broken apart, or they'll be put together. So we have some words we're going to learn. Substrate. Substrate is the molecule that the enzyme is acting on. This is like our key. Okay, so the substrate's our little key that's going to go into the enzyme. It's going to bind or connect to the active site. Think of this as the keyhole, right? This is where we're putting the key in. So the active site is like the keyhole. And they have to fit together. They have to have the right fit. If this guy's the wrong shape, it's not going to be able to bind to this active site, and it's not going to be able to break apart. So they must fit together. So our key and our keyhole have to fit together. Here's another example, right? So our key here, there's two keys. You got to put them both into the keyhole, the active site. All right. And then eventually, right, they're going to form our product. Thing here, right? Our product, our end result. So this lock and key model explains how an enzyme works, kind of like a key and a lock. The substrate is our key, has to fit into the active site, the keyhole in the enzyme. So the active site's in the enzyme. And then once it fits in there, if it's the correct shape and we're working at the correct temperature and pH, then we're going to produce our products.